Hello everyone. In this video, we are going to see what is CRISPR-Cas9 gene editing. CRISPR stands for clustered, regularly interspaced, short, palindromic repeats. And Cas9 is basically a protein which is seen associated with the CRISPR, and the expansion of Cas9 is CRISPR associated protein 9. From the name itself, we could interpret that CRISPR is a DNA sequence and Cas9 is a protein which is seen associated with the CRISPR. Gene editing is a revolutionary technology that allows scientists to make precise changes to the organism's DNA. Before we discover CRISPR technology, scientists used two gene editing techniques called zinc finger and talons, and it paved the way for CRISPR-Cas9. CRISPR-Cas9 is basically uh, a part of immune system which is seen in bacteria. So we are going to see what exactly is CRISPR-Cas9 and how scientists have discovered it. Imagine this as a bacterial cell, and this bacterial cell is getting attacked by a phage. A phage is nothing but a virus which infects the bacteria. So this phage is attacking bacteria. When this phage attacks the bacteria, it transfers its own genetic content to the bacterial gene pool, and this gene will try to integrate with the bacterial genome, and this causes the infection. But scientists have observed that when the viruses transfer their genome into the bacteria, what the bacteria does is bacteria will take a copy of the viral genome and using transcription they will generate a RNA copy of the DNA, and the Cas9 protein will come and attach to this RNA. This RNA is called cRNA or CRISPR RNA. So what's gonna happen next is, imagine the same bacteriophage is attacking the virus bacteria again. The same virus is gonna attack the bacteria again. Now what is gonna happen is, like before, it transfers the DNA into the bacteria, but now we have the cRNA associated with Cas9 protein. So these two sequences are complementary to each other. The viral DNA sequence and the RNA sequence we have are complementary to each other. So what happens is a bond is formed between this viral DNA and our RNA, and this RNA basically guides the Cas9 protein towards the viral DNA. Now what will happen next? Since it is both the sequences are complementary, they will form a RNA DNA hybrid. As soon as this RNA DNA hybrid is formed, the the Cas9 protein, which was attached to our RNA, will cleave the viral DNA and destroys it, tries to destroy it. Now let us see what will happen next. So this is basically what I have told you right now. The bacterial uh, genome will come and attach to the cell and cell's genome, and the CRISPR RNA along with Cas9 will come and destroy the viral sequence, leaving a very large space in between two DNA. So this is the mode of action. So since the RNA sequence is DNA sequence is destroyed, the cell will try to fix this sequence. That we will discuss later. Now let us look at the mode of action of CRISPR. So CRISPR-Cas9 gene editing operates through a multi-step process involving intricate molecular interactions. The first one is acquisition of spacer sequences. So as I have mentioned earlier, the viral DNA, when it enters the cell, the bacteria will capture a copy of the viral DNA, and through transcription it will synthesize the corresponding RNA sequence, and the Cas9 protein will come and attach to it. Next is the transcription processing. This is that part. The DNA, the copy of DNA is made. That is the first part, acquisition of spacer sequence, and the next one is transcription and processing. The RNA is produced, and the third one is formation of ribo nucleoprotein complex. So the RNA is produced, and the Cas9 will come and attach to that RNA sequence. Next is target recognition and binding. When when the same virus enters the cell and it transfers the same DNA, then the cRNA, that is our CRISPR RNA, along with Cas9 protein, is gonna recognize the target DNA, and this RNA will lead our Cas9 to the DNA, and DNA RNA hybrid is formed. As a result, DNA cleavage, the bacterial DNA is cleaved. So as I have mentioned, if it is cleaved, the bacterial whichever cell it is, it will try to fix it. So Next is DNA repair. The cell's repair machinery can either introduce small insertions or deletions via non-homologous end joining, or incorporate desired changes through homology directed repair using a donor DNA template. So what is going to happen here is, um, in the first one, non-homologous end joining, the DNA is cleaved, this much portion is left, so the cell will try to add nucleotides to this location to fill the gap. But this isn't a very precise technique, because it may add some random nucleotides, and this will lead to mutations, and this may lead to mutations. It will not necessarily lead to mutations, it may lead to mutations. And the next one is homology directed repair, the second one. This is how we exploit the power of our CRISPR Cas9 gene editing. So our CRISPR is here. This CRISPR is gonna cleave this much of portion. And imagine I want to create a particular DNA sequence of in this portion. So what I'm gonna do is, I will provide a complementary DNA sequence over here. So basically I'm telling the cell that I will provide a complementary sequence over here, and you can add new base pairs according to this sequence. So I'll tell you what to do. You just add the nucleotides. That is what is going to happen here. The cell will identify the complementary sequence and add the nucleotides according. So my gene of interest has been created over here. So I haven't isolated any gene, I haven't transferred anything into the host cell. I have just used the CRISPR and transferred and provided a complementary DNA sequence, and the cell itself created the DNA sequence which I wanted. So that's how we use the power of CRISPR. Now, as I have told you, CRISPR is a very versatile gene editing tool composed of two main components, that is CRISPR RNA and Cas9 protein. So it works by guiding Cas. It works by so it has a guiding strand, that is the RNA strand, plus CRISPR. Plus Cas9 protein. That's how CRISPR works. Uh, CRISPR RNA contains a sequence complementary to the target DNA. Transactivating cRNA assists in the formation of a functional complex with Cas9. Cas9 protein is an endonuclease that cut DNA at specific locations guided by the cRNA. So Cas9 protein will not cut any random sequence. That sequence should be similar to should be complementary to the sequence of the RNA. Then only it is gonna cut. And Cas9, it is an endonuclease. CRISPR-Cas9 gene editing is a revolutionary technology that allows scientists to edit the DNA of living organisms. CRISPR as I have mentioned earlier, CRISPR stands for clustered regularly interspaced short palindromic repeats, and Cas9 stands for CRISPR associated protein 9, and it is basically an enzyme that plays a key role in this system. So, uh, this is the Cas9 part, and this is our RNA. So this RNA attaches to the viral DNA, and Cas9 cleaves that portion, creating this gap. So now we are going to look at the applications of the CRISPR-Cas9 technology. CRISPR CRISPR-Cas9 enables a wide range of research applications, including gene knockout to study gene function, gene knock-in for precise genetic modifications, gene regulation to control gene expression, and functional genomic screening to identify gene functions. In medicine, CRISPR-Cas9 holds promise for treating genetic disorders by uh, correcting disease causing mutations, cancer therapy, and targeting cancerous cells, and infectious disease research, such as developing HIV resistant cells. Now, let us look at the recent advancements and future directions. Recent advancements include the development of improved CRISPR variants such as base editing and prime editing, addressing challenges and opening new possibilities for applications in various fields. Future research aims to refine techniques, address safety concerns, and explore novel applications. Now, let us see the ethical considerations. Despite its potential, CRISPR-Cas9 raises ethical concerns, including target effects that may lead to unintended consequences, germline editing with implications for future generations, equity and accessibility of treatments, and the need for robust regulation and oversight. So the conclusion is that CRISPR-Cas9 represent a groundbreaking technology with the potential to revolutionize uh, genetic engineering and medicine. While ethical considerations and technical challenges remain, ongoing research and advancements continue to expand its applications and impact. So this is all about CRISPR, and I hope you have enjoyed the video, and thank you.